Parrish, nice to see you again.
Hi, thank you, Ben, and thank you everyone for joining. So I'm going to talk about tech options to improve low productivity biotherapeutic process. So this story is actually comes from my distant past, in which it was a small biotech startup company, and the company was in a race with a competitor to bring a drug to clinical trials for treating AIDS patients. And our competitor had very deep pockets with regard to process development expertise and familiarity with cell culture. And that was in contrast to our company, which had very little experience way back then. So we put together a plan to bring this drug to the clinic and took it to exec management. And naturally, exec management said, sorry, you don't have a big enough gun to your head here. You need to get that drug to the clinic more quickly than what you've been saying in your current plan. So we had the other challenge that the cell line that was available was a low producer. In batch culture, it only made 6 milligrams per liter of product, which was incredibly low. And back then, it even required serum. So we scratched our heads and decided to go with a higher productivity process, which was perfusion. And we went back to the exec management and proposed that. And they said, okay, why don't you go ahead and start it up? So we worked with an outside company to bring in perfusion technology. And we installed it in the facility. It was stainless steel based, so there was no single use involved back then. And that made it doubly difficult to install the systems, which had to be steam traced and cleaned in place. And also, we didn't have we had a small process development group, so there was very little resources available to model the process in the lab before moving it into pilot or clinical production. So those were the challenges that we had. So we ended up deploying the technology in the production facility without a lot of process development backup. And as a result, we had a lot of difficulty, or we experienced a lot of difficulties in installing the systems, getting them validated, and starting them up and getting them into production. In parallel, we decided that we had to make an investment in process development. So we did hire some people to come in and develop a small-scale benchtop system that could be operated to simulate the process and to model it so we could find out ahead of time how this process was going to perform. So we began production in our facility, and these processes were designed to run very long. So inoculation, grow up in the culture in the system would take about 3 weeks, and then it would grow for, we would continuously produce product for a number of months using the system. And we found out, luckily, that the titer increased tenfold compared to batch production in this continuous system, due to the fact that we developed very high cell densities on the order of 200 million cells per mil. This was a system that used microcarriers, so the culture was immobilized in those microcarriers. And that enabled us to do purification more easily because we did not have so much challenge removing the cells from the cell culture harvest. So we got into production with that system, and we installed a second system. So we had 2 systems running in parallel. and each system consumed 20 reactor volumes per day of media. So it turned into a media production operation and a harvest operation to beat the band. And so every day was a real mill of making media and harvesting cell culture fluid on the backside and getting it over the capture columns. So production started pretty well after we got all the startup issues worked out, and we got into regular manufacturing. We accumulated quite a bit of product and put it in the inventory. And it turned out that we got into the clinic ahead of our competitor. So that made executive management very happy. Another challenge came in that we did not have enough experience to predict what would happen if anything went wrong with the production systems once it was up and running. So, for example, after 4 months of running one of these systems, I got a call from QC saying that the product was contaminated with endotoxin. And so the question was, what are you going to do about it? Well, I ended up sitting in a room for about a month with the quality assurance organization, who rightly pointed out that we could not go back and figure out exactly when the system got contaminated. It was not a rampant contamination, so it was sort of cohabitating with the cell culture process. But we could not, because of sampling insensitivity, that is, when you sample a system, you're only sampling a small portion of the reactor. We could not guarantee when the reactor became contaminated or when it was cleaned. So unfortunately, the downside was that we ended up quarantining 4 months of product that we had produced while we argued with QA about the ultimate destiny of that product. And that was a very expensive endeavor in time and effort, for sure. So we ended up meeting the goal of getting the product to the clinic ahead of our competitor, but we ended up with a very protracted manufacturing arrangement with quality assurance. In fact, a very unsure footing with continuous manufacturing, especially trying to manufacture a system or run a system for such a long period of time. So the lesson that we learned was that we did not have an agreement with QA about a downside scenario of a potential contamination of the system and what would be the operational outcome or action plan if that did occur. And the bottom line was the quality assurance group required that we, in fact, dump the batch of 4 months worth of product, which was to the tune of millions of dollars of cell culture media cost and time and labor in the plant. Now, in retrospect, that soured the opinion of many people in the company about the viability of producing things using continuous culture. So for our next product, we ended up scrapping the perfusion system, ripping it all out, and replacing it with a fed batch facility that we then installed and started up. So overall, it was, yes, we got the drug to the clinic, but it was a very painful learning curve on the logistics and the arrangements between quality assurance and operations for how to manage a downside scenario with a continuous operating system.
Yeah, so 20 VVDs, that's something that was written in the chat box, and I agree, that's a tremendous amount of volume. Do you find, in retrospect, that you think that the logistics, the throughput and logistics had anything to do with why the technology was scrapped, or was it really all about the dollar value of the contaminated discard?
Well, I think it was both the dollar value, but also the lack of assurance of the validity and the robustness of running continuously, and what it takes operationally to maintain that in working with QA and maintaining product quality for such long periods of time. I think today, continuous manufacturing in the upstream is only done for a couple of weeks, or maybe 3 weeks, typically, or in most companies that do it, because of the risk of contamination and the inherent media cost and labor costs associated with a downside failure scenario. So to answer your question, I think it was a combination of the logistics of maintaining a continuous operation, running at that rate, and the cost incurred by losing, months worth of product.
Yeah, that sounds pretty terrible. I guess, maybe some parting thoughts you might have for the audience as to any other major barriers that in hindsight stick out in your mind that others should be on the lookout for?
Well, certainly if you're considering continuous manufacturing, you need to put some money into, or a fair amount of investment into your process development organization to give them the tools to fully evaluate the technology and to assess the productivity and risks of going that route versus alternatives, batch or fed batch alternatives. And that's an area that's frequently not funded very, very well in biotech companies these days. So that would be my advice.
Yeah. No, I think that's pretty salient advice, definitely. And, have you seen anything to that point? Have you seen anything in your mind's eye change as time has gone on, where you think back and what your immediate reaction was now seems a little bit softened or different in some way?
Well, I think it's softened by the fact that the productivity improvements with continuous manufacturing in the upstream is quite significant. I mean, bioreactor productivities can be improved 3 to 6-fold by applying perfusion technology to the bioreactor. And that has had a major impact on the size of facilities and the cost of goods as well as the plant output. So I think that's what's changed over the years. Plus, the technologies for doing continuous have improved as well.
Yeah, absolutely. And thank you for that, Parrish. That was an excellent case study, and I appreciate hearing a lot of the background and how you were able to bring your expertise to bear there.